Here we go, embarking on Chapter 3 in the Zimmer and Emlyn evolution textbook. So Chapter 3 is titled, What the Rocks Say. Subtitled, How Geology and Paleontology Reveal the History of Life. So here's the thing. The Earth is very old. How do we know this? Geology. And life on Earth is very old, and there's a pattern of transition over the history of life on Earth. How do we know that? Paleontology, the study of fossils. So both of these fields, geology and paleontology, reveal to us the history of life on Earth. And that's what we're going to investigate in this chapter. Now, by learning about these topics, about researching these areas, we can learn mechanisms of evolution better. We can document the emergence of new species and learn how that may happen historically. We can see the extinction of major lineages over the history of life and examine what caused those extinctions. We can even come to understand ecological relationships. We can reconstruct ecosystems from fossils in the past. And by doing this, we can illuminate the emergence of ecological partnerships. So even though we're talking about things that are dead and have been dead for a long time, we can creatively and intuitively come up with ways to learn a lot about life in the past. Now, the image that's on the bottom right-hand part of your slide here is of some living stromatolites. Stromatolites are layers of rock or rock-like material formed from bacteria, biofilms of bacteria, most especially cyanobacteria, but other types as well, living in colonies in biofilms. So these stromatolites exist in a few rare places on Earth today, but some of the earliest fossil life on Earth is of stromatolites. So these unlikely formations and unlikely creatures actually give us a window into the past for the beginnings of life on Earth. So how do we know how old things are? Of course, this has been a obstacle for scientists for hundreds of years. And only relatively recently, just a little over 100 years ago, did we really develop ways to pinpoint with great accuracy how old rocks are on Earth. And the methods that we use to do that fall under what's called radiometric dating. And we'll get to that on the next slide or two. But before radiometric dating, how did geologists estimate the age of the Earth? Well, basically they were left to studying how long it took for, say, example, sediments to accumulate in riverbanks or in coastal waters, and based on the rates of accumulation of those sediments, they inferred how long it would take to make the layers of rocks that we see in the geologic record. So not super reliable, but those methods were beginning to suggest that the Earth was much, much older than what, say, the biblical creation stories said. These geologic methods or methods of geologists suggested that the Earth was tens of millions of years old. Now, Darwin, he actually made a contribution to this in England. He applied techniques to estimate the age of the Weald. That's an area of England. It's a stretch of ridges and valleys in southwest England. And that's what the map on this slide is showing you, this region called the Weald. So he applied these techniques that geologists use, and he estimated that this region would be up to 300 million years old. And he then went on to say that, hey, if this region is 300 million years old, then certainly the Earth has got to be much older than that. So even in the 1800s, geologists, and Darwin did work with geologists, had come to conclude that the Earth is hundreds of millions of years old. In fact, from these estimates, Darwin was even suggesting billions, and he was correct. So this is how early geologists attempted to identify the ages of various formations on Earth, was by sort of the uniformitarianism principles of Lyell, using measurements today to see how long it takes for rock formations to form, and then inferring in the past how long ago those events must have taken place. Now, a contemporary of Darwin's was Lord Kelvin, a physicist, and among his many contributions to physics, he pioneered research on the flow of heat through solids. And so, based on what he had learned through the mechanics of heat transfer, he inferred that the Earth must have started off first in its beginnings as a molten blob, then cooled gradually, forming a crust on its surface, a solid crust, but then the interior of the Earth would have remained molten. And since rocks cool at a steady rate, he examined rocks deep inside of mines to ascertain what their temperatures were and what their heat transfer was. And based on these measurements of rocks and mines, and the reason why he did mines and not on the surface is because the surface is continuously warmed up from the sun and then cools off, at night. So those measurements indicated that the Earth was no more than 20 million years old. Now, that would be a problem for evolution, because the complex life forms that are around on Earth today would probably need longer than that to originate and come about. So now Kelvin's work had a problem, had a flaw in it, and Darwin couldn't figure this out at the time, but Kelvin assumed that the Earth was a rigid sphere. But what he didn't know is that the Earth has a molten interior that rises towards the surface, cools, and then sinks again towards the interior. So basically, the inside of the Earth is turning over, kind of like taffy, kind of gooey, and the upper layers are warmer than in the Kelvin model. So because of this turnover of the Earth's interior, it gave an incorrect age for how old the Earth is. 20 million years was just too young. So now we turn our attention to radiometric dating. And the title of this section is A Curious Lack of Radioactivity, which you'll see what that title means in just a moment. But we first need to review that in the world of chemistry, in our periodic table of the elements are represented by atoms, and atoms can either be stable or radioactive, unstable. So atoms that have equal numbers of protons and neutrons in them are stable and are unchanging. Atoms that have unequal numbers of protons and neutrons are unstable and radioactive. Some of those atomic particles, subatomic particles, can decay in the proton-neutron world, turning that atom into something else. Now, we can use this information about elements decaying and turning into other elements, which is a property called half-life. So those radioactive elements, the different forms of elements, by the way, are called isotopes, but those radioactive isotopes can decay and turn into other things. So by measuring the proportion of radioactive isotopes to their daughter isotopes, the parent isotope to the daughter isotope, we can infer how old a rock is. So this is all based on physics. Look at the table that I've inserted here. It's giving you some examples of radioactive parent isotopes, and then showing you in the middle column the product, the daughter isotopes that form from them. And so, for example, samarium-147 has a half-life of 106 billion years. It takes 106 billion years for half of a sample of samarium-147 to turn into neodymium-143. If you look down farther in the table, all the way to the bottom, we've got carbon-14. And carbon-14 is a radioactive isotope, and it decays and turns into the daughter isotope nitrogen-14, which is more stable. And that only has a half-life of 5,715 years. So you can see in this table that physicists have figured out a wide range of half-lives that exist for different radioactive isotopes. So as I was saying, if we can measure the proportion of the original isotope to the daughter isotope and how much is in a rock sample, for example, we can estimate how old that rock is. Most of these radioactive isotopes were synthesized in ancient stars long ago, and they were present upon Earth's formation. An exception would be carbon-14. Carbon-14 is continuously replenished up in our upper atmosphere, and so that is relatively stable. Now we're getting near the bottom of this slide. All the discovered isotopes have half-lives over 80 million years. So what this implies then, getting to the title of this section, A Curious Lack of Radioactivity, is there's an absence of radioactive isotopes that decay relatively fast. There is a complete absence of radioactive isotopes that have half-lives that are less than 80 million years. So what this tells us is those radioactive isotopes have all disappeared. The Earth, the universe are too old for those isotopes to still exist. So that is consistent with the ages, the long old ages of the Earth and the universe in general, is the fact that these isotopes with shorter half-lives do not exist anymore. So now we get to the geologic record, and the geologic record is basically a table. It's basically a figure that geologists put together mostly in the 1800s through surveying. And what the geologic record shows is the major transitions in life's history that we've learned from the fossil record. And we have dates on those transitions, and those are the numbers that you see in this figure, are the numbers of millions of years ago that these transitions took place. So the transitions are identified by the fossil record. We can tell by looking at the layers of rocks, from the younger rocks down to the older rocks. We can tell what the transitions were and where they were in the layers of strata of rock. So we can identify how long ago those transitions took place. Those are the numbers of years, millions of years, by using radiometric dating. So what geologists do is they date rocks that are below and above a fossil. And based on those dates of the rocks that are below and above, they get an estimate of how old that fossil must be. That's how we arrive at these data. Now, the reason why we can't just date the fossil is because fossils are found in sedimentary rock, and sedimentary rock rock is made up of all kinds of particles from different kinds of rock. It doesn't give an age that that sedimentary rock formed. But volcanic rock, rock that comes out of volcanoes, lava rock, that can be reliably dated. So what geologists are doing to get ages of fossils is, like I mentioned a moment ago, dating the volcanic rock above a fossil to get the younger age, and dating a volcanic rock below a fossil to get the older age, and then having a bracket to get an idea of how old that fossil is. So this is how we get the geologic record. So this section is titled A Vast Museum, because indeed the fossil record is a vast museum, a huge collection of artifacts and remnants from past life that gives us this history of what has existed before us. Now, critics of Charles Darwin, he had a whole chapter in his Origin of Species book about fossils, and he published other things about fossils. But some of Darwin's critics said, you know, the fossil record is incomplete. It provides an incomplete chain of fossils from one form to another. There are fossils missing that should be transitionary. And given that there's fossils missing that would be transitions between one thing and another, they argued evolution isn't supported. Well, Darwin's explanation was, hey, give me a break. The fossil record is imperfect. It's not a perfect record of the history of life. It's imperfect. And it's imperfect for a number of reasons. Let's document a few. One of the reasons is because not all organisms fossilize easily. Most organisms that fossilize have hard parts, like skeletal parts. Sometimes types of organisms just don't have hard parts, so we're not going to see fossils of those. We're not going to see transitions of those. Another component is fossilization takes a very specific kind of environment. An organism has to become trapped in just the right kinds of sediments to turn into fossils. Well, many life forms don't live in environments where fossils would form easily. So there's another reason. A third reason is the oldest fossils on Earth have been destroyed. They become destroyed because rocks are eroded, landscapes are eroded by rain and wind, and the Earth's crust keeps breaking down and reforming. So fossils that formed hundreds of millions of years ago, there's a less likely chance that they're even still around today. So there's a lot of reasons that Darwin identified, and they were accurate. They're correct. But since Darwin's time, those critics had an argument that there were a lot of transitional fossils absent. Well, of course, we've discovered many, many more since then. Now, how do organisms become fossilized? This diagram here on the slide kind of gives you a pathway, a picture of what happens. So, of course, first you got to have an organism die. It might be buried under a pile of sediments and die that way, or maybe it dies naturally and then it gets buried afterwards. But one way or another, it dies, and it has to be buried before it decomposes. So if something's dead on the surface of whatever, the seafloor or on land, lots of scavengers and predators can eat it and pick it off before it has a chance to get buried. So it's got to get buried. Now, if the remnants, like hard parts, bones, etc., end up getting buried, then there can be a slow, gradual process of the actual hard parts, like bone or a shell, being replaced with minerals, surrounding minerals in the rocks and soil. So the original material dissolves away, and it leaves the new minerals in its place that represents the form and shape of the fossil. So it's a very common way for fossils to form, but it's not the only way. Now, in order to be discovered, the rocks have to be lifted up from lower layers, and the overlying layers need to be eroded away from water or wind so that we can then see the fossils that formed long ago. So we have to be pretty lucky It's the bottom line, to happen to find a fossil when it just happens to be revealed from erosion and uplift, and to find that fossil before it's destroyed by the elements. So despite the fact that the odds are against a lot of things fossilizing and being preserved for long periods of time, we do have a pretty good record of the history of life, as we'll get into further in this class. Now here's an example of using the radiometric techniques and other techniques I've just described from your book to date how old a human fossil is, specifically from our species, Homo sapiens. So this fossil was found in the Omo River Valley in southern Ethiopia, and of course on the surface of the ground or digging in a hillside. And so we're left to ask, well, how old is this Homo sapiens fossil? As I've talked about, we can't directly date the rocks that the fossils are found in. And the reason for that is because the fossils are found in sedimentary rock that's formed from sediments of mud and sand particles packing together. And those sediments are from many, many different ages of rock. So that's not going to work well. Instead, as I mentioned, we need to date the volcanic ash layers or the volcanic rock layers above the fossil and below the fossil to get the younger age and the older age. In this case, the potassium-argon method was used. So radioactive potassium decays into argon. And we know the half-life. We know how long it takes for potassium to decay into argon. So by looking at the ratio of those isotopes in the volcanic rock layers, we can estimate how old those volcanic ash layers are. So by doing that, it turns out that the upper layer is about 104,000 years old, and the lower volcanic layer is about 196,000 years old. So that means that Homo sapiens fossil is between those ages. Now, because the fossil was found very close to the lower volcanic rock layer, it stands to reason that it's not much younger than the oldest rock. So it's been dated at, maybe around 180,000 years old or so. So this is providing a good example of how we date fossils, how we age them. So here we can see that fossils are not just showing us what kinds of life forms existed in the past. I mean, clearly that's a major goal, is to know that information. But we can learn a lot about the biology of past life forms based on the remnants that have been left behind. So we can learn lots of different kinds of things besides just the existence of a species. Behavior. We can see behavior being fossilized. So the upper image on the right-hand side of your slide shows a couple turtles that were getting it on. They were mating, and they got buried and killed and fossilized. So there we have fossilized mating behavior. Fossils can show us the development of organisms, of animals, and we can have an idea of how fast they grew and the different stages in their lives, etc. So on the bottom of the slide, it's showing you these animals called trilobites that are arthropods, and we have fossilized remains of many different sizes of them, of different ages. And so there's a graph down there on the bottom that's showing you how we can plot the total cephalic or head length against the maximum cephalic width. And by doing that plot, we can see the rate at which these guys grew and developed. So we can get a sense of their development. We can also see something like predation. Look at the bottom image on that figure to the right. There's a fish eating a fish. The middle image I haven't mentioned yet is an ichthyosaur, a fossilized marine reptile, that was in the process of giving birth and shows some embryos or fetuses inside of it. So we have an idea, we have an understanding of what kind of reproductive strategy the ichthyosaurs used. So the overall point here is that fossils do more than just tell us that a species existed, but we can actually learn about behaviors, growth patterns, ecosystems, all kinds of things like that. So bringing fossils to life. So this is pointing out several examples of how we can come to understand aspects of these animals' lives by studying the fossils. Again, another testament to we're not just finding out what species existed in the past, but we can come to understand the details of their biology, including behaviors. So for example, Tyrannosaurus rex, we're able to estimate how fast they were able to run by developing biomechanical models. Physics, it's totally physics-based of biomechanical models of running, looking at living animals. So we can look at, say, for example, alligators and birds. They are the closest relatives to Tyrannosaurus rex. And we can figure out biomechanically how they run, how they move, look at the anatomy of a Tyrannosaurus rex, and what was concluded and published is that Tyrannosaurus rexes were not very good runners. So as predators, they may not have been that great at running after and grabbing their prey. They may have employed some other strategies, like maybe they were more scavengers. Anyway, by knowing something about their running speed, we can learn a little bit more about their biology. Scanning electron microscopes have allowed us to look at the intracellular structures of fossilized birds. This animal called Anchiornis, which is an ancient early bird. We're able to see using electron microscopes melanosomes. So those are structures in cells that produce pigment. And by us being able to identify these fossilized melanosomes, we can infer what colors these animals were. The bottom bullet is talking about CT scans, CAT scans, and how they allow the view of internal structures like the head crests on a hadrosaur, dinosaur. Pretty dramatic sort of structure on their heads. What were these head crests for? Paleontologists have come up with several hypotheses of what they're for, but the evidence suggests that they were for vocalizing, bellowing, to communicate with each other. And so some of this evidence that supports this comes from the fact that the ear structures of hadrosaurs were tuned to hear the frequencies of sounds that the head crests would have been able to produce. So that's a consistent model that suggests to us that hadrosaurs used that bizarre head crest to make sounds and vocalize for communication. Bringing fossils to life. Pretty cool stuff. So here's a wee bit more about bringing fossils to life. Lagerstätten. So there should be two little dots above the A in that word, umlaut. Fortunately, I took German in high school, so I know that that would be pronounced Lagerstätten. So Lagerstättens are examples of fossil-rich sediments that basically show us the whole entire ecosystem, like the whole range of living things that lived in this ecosystem were fossilized quickly and provide a very vivid snapshot or glimpse into past life. Lagerstätten. So typically this happens when animals are swept into anoxic pools, that is oxygen depleted pools, lagoons or bays. So organisms are unable to, like microorganisms are unable to decompose the dead things. And so everything gets preserved because it's not being decomposed and broken down. So these anoxic, oxygen poor environments are protecting these dead creatures from bacteria and scavengers. One premier example of a Lagerstätten is the Burgess Shale in British Columbia, Canada. So it's in the Rocky Mountains of British Columbia. So if I remember correctly, it's up around 7,000 feet elevation. And there's this rich fossil deposit in areas of the mountain range here of marine animals, ocean animals from 520, 510 million years ago. And it's a Lagerstätten because there is such a rich assemblage of fossilized animals that apparently they got buried underwater, under the ocean, when hillsides broke off underwater and tumbled down, taking the organisms down and completely burying them. Everything was preserved. So 65,000 specimens have been collected and described of more than 93 species, and it helps us to develop a picture of an entire ecosystem. That's a Lagerstätten. So traces of vanished biology. We even have fossilized molecules that can help us to understand life in the past. So coal of what we burn as a fossil fuel, was formed when plant matter was buried under deep layers of sediment and pushed deep down into the earth, and under tremendous pressure, that organic substance was compacted and converted into coal. Now, coal balls are little spherical structures that we occasionally find within coal that has like fossilized parts of plants that are still in there, little stems and leaves. So that's that's not a molecular kind of example, but an obscure place to find fossils for sure. Biomarkers, these are the organic molecules that I'm referring to. Organic molecules formed by biological processes. And so by identifying these biomarkers, we can learn something about life in the past. Okanane, that is an example of a biomarker that's only produced through a series of specific enzymatic reactions, in purple sulfur bacteria. So it's very specific that these organic markers are only made by purple sulfur bacteria. So we have found fossilized okanane dating back to 1.64 billion years ago, and that tells us about the chemical environment of the ancient oceans, because these purple sulfur bacteria are anoxic, they are anaerobic, they do not thrive well in oxygen-rich habitats. So if we detect these okananes, that means we are detecting sediments from where bacteria lived, purple sulfur bacteria, and they would have been very oxygen-poor environments. Heavy versus light oxygen isotopes. Well, this was pointed out in chapter 1 in our whale and virus chapter, that there is oxygen 18, oxygen 16, with 18 being heavier, and these isotopes are in different proportions in salt and fresh water. So this isn't an organic marker, this is an inorganic marker, but we're finding it in fossilized whale teeth. And based on the ratio of those isotopes, we know where those whales must have lived. So those are biomarkers. C4 plants and C3 plants. This has to do with different types of photosynthesis. C4 plants are essentially grasses, and they have much more carbon-13 than C3 plants. So by measuring the proportion or amount of carbon, the type of carbon isotope in a fossil, that tells us what kind of plant that animal must have been eating. A plant that was more of a C4 plant, like a grass, or a plant like a C3 plant that maybe was a fruit-producing plant. So the example to your right is showing you what we've learned by looking at some carbon isotopes in fossilized humans and in other primates. And we have the age on the x-axis in millions of years, and then on the y-axis we have the spectrum of, near the bottom of the y-axis, carbon-3 plants, so being rich in carbon-12, and then carbon-4 plants up on the top that are more rich in carbon-13. And what we can see from this, Homo sapiens are way over on the right because we are recent, okay, in that time scale. And we are, you can see we're more in the C3 plant range. So we're not eating grasses, we're eating, fruits and various other vegetables. But you can see the purplish dots that are kind of high up in that graph. Those belong to a genus of early human called Paranthropus. And they had a higher signature of carbon-13, indicating that they're eating more C4 plants like grasses. And in fact, their teeth were modified to be more of a grinding type tooth that was probably adapted for eating grasses. So the point here is, again, we can use the biomarker of the type of carbon isotope that's in the fossil to ascertain what kind of plants the animal was eating. Pretty cool stuff. Traces of vanished biology. So we're going to take a quick look at Earth's history here. Reading the record, beginning with early Earth. So using the techniques we've talked about in this chapter with radiometric dating of meteorites and the oldest rocks on Earth, the Earth formed 4.5 billion years ago. Actually, to my knowledge, there are no rocks on Earth rocks that are that old. They've all been destroyed through geologic activity. So the oldest rocks are from meteorites, moon rocks, for example, and these other samples help get us to this 4.5 billion year age. So the model of how our Earth then proceeded from that point on is it started off as a molten ball, would not have wanted to be here, could not have been here, and it began to cool and form a crust, so the outer covering hardening like an eggshell. Gases were escaped to form the atmosphere from the geologic activity that was occurring on the surface. Water arrived. The model for that is water was brought in from comets extraterrestrially. There's abundant water, really, in our solar system. And that water, as our Earth began to cool, the water condensed and began to rain. This started to produce oceans. 4.4 billion years ago is when our moon formed. And that was quite a contentious debate for many years. I recall during my lifetime of some hypotheses going back and forth. But most recent evidence supports the idea that early Earth was hit by something massive, perhaps close to Mars-sized planet, and literally, ejected out a huge mass of material from our planet that formed into our moon. Quite an image. The giant impacts from asteroids, meteors, comets started to taper off about 3.8 billion years ago. And tectonic plates formed in our Earth's crust and began moving around. So the formations of continents and other land masses that are then moving on the surface of the earth. Erosion from rainfall and wind, but especially rainfall, and plate tectonics continued from then on, and older rocks continued to be destroyed, replaced by newer ones. So the farther back we go in Earth's history, we find fewer and fewer old rocks. They've been destroyed by various geologic processes. So we don't have much left from the earliest days of our Earth. In fact, as I said above, we don't even have rocks that date back to 4.5 billion years ago. The oldest ones, I think, are about 3.8 billion years ago. So let's continue with our quick summary of Earth's history. So, life's early signatures. What is some of the earliest evidence we have for life on Earth? Now, life uses a lighter carbon isotope, carbon-12, and so that's one signature that we potentially can get from molecules, as if they're rich in carbon-12, then we might be seeing signatures, biomarkers, if you will, for life. So the ratio did shift 3.7 billion years ago. So it's suggestive that life may have been around then to shift that ratio. But the good thing about science is that people are skeptical until evidence accumulates. Critics argue that geologic processes could have produced the same result, so it's controversial. But the oldest fossils that we have of life that are not disputed date back to a little older than 3.4 billion years old. Stromatolites, which I'm picturing here on the slide. Some contemporary stromatolites in shallow tropical seas, and then the image below fossil stromatolites, such that if we did microscopic examinations of the fossils, we would see cellular structures from bacteria. Now, I put the question mark next to 3.4 billion years ago because I was wanting to remind you or ask you, how do we know that these things are that old? How do we figure that out? It goes back to earlier in the chapter with radiometric dating. So stromatolites were very abundant before 541 million years ago. I don't remember why I had that question mark. Now, living stromatolites are only found today in salty, saline lakes or shallow lagoons. They're not very common. There's only a few special places on Earth where we can find them. I've never seen them in person. That would be a neat thing from a biologist, evolutionary biologist perspective to see with my own eyes. Another landmark here, if you will, for our early signatures is atmospheric oxygen began accumulating 2.4 billion years ago. So what would that oxygen have accumulated from? This is a question to you. You might remember back from something you've learned in Bio 182 even, because that's important background. So what kinds of organisms are releasing oxygen into the atmosphere or into the water? And of course the answer is photosynthetic organisms. Now, 2.4 billion years ago, that is before eukaryotic life was around. So what kind of life forms would have been around 2.4 billion years ago? Bacteria and archaea. Well, what primary type of bacteria does photosynthesis? Cyanobacteria. And cyanobacteria, the fossil record shows that cyanobacteria arose close to this time. Coincidental? Maybe, maybe not. But the oxygen probably was coming from cyanobacteria. And once the oxygen was accumulating in the oceans from photosynthesis, it could start leaking into the atmosphere and altering Earth's atmosphere, changing its chemistry. So this section is titled The Rise of Life. The last section in the slide was about detecting early life. So now we've detected it. How is life rising up? This figure, you'd have to go to your textbook to read it carefully. The font's pretty tiny there, but it's showing you a network-like diagram for evolutionary relationships of bacteria and archaea and eukarya. So you might remember from Bio 182, and I don't know what other upper division classes you may have had since then that would touch on these kinds of things, perhaps not any. But you might remember that archaea and eukarya are hypothesized to be closely related. In fact, recent evidence shows that eukarya evolved within the archaea. So we are, in fact, archaeans. But anyway, Precambrian paleontology, the Cambrian period began about 540 million years ago. So fossil history before that is challenging, mostly because we don't have a lot of fossil evidence. So how do early fossils relate to modern life? Well, of course, we all share a common ancestry. So early fossils are connected to us genetically. And we have these three domains that I've just been talking about, the bacteria, the archaea, and the eukarya. Now, the cyanobacteria I have here date back from 2.6 billion years ago. Pretty close to when we started seeing atmospheric oxygen start taking off. So significant why? We've just addressed that because they are photosynthetic and they release oxygen so they can start changing the planet's chemistry. So oxygen levels remain low for quite a long time, and purple sulfur bacteria remained common for a long period of time. How do we know that? Remember those biomarkers earlier, like the ocanines that are produced by the purple sulfur bacteria. So we know this from those biomarkers, that paleontological chemistry. Methane has a relatively low amount of carbon-13, is found in 3.5 billion-year-old rocks. So that might be traced to Euryarchaeota, a type of Archean, but then maybe it's non-biological. We're not quite sure. Eukaryotes, we see the first fossil eukaryotes show up about 1.8 billion years ago with definitive eukaryotic cells. They're about 100 times bigger than the previous prokaryotic cells. So cyanobacterial symbionts gave rise to which important organelle in photosynthetic eukaryotes? The chloroplasts, of course. So that's an important early event. With the origin of eukaryotes, we begin getting the origins of predators too, because the relatively large cells are becoming multicellular. So the world's changing. It's taking a long period of time, right? Hundreds of millions of years for these transformations, but the world is changing. And the world at that point was still dominated by microbes, bacterial and archaean microbes. So at some point, life starts getting bigger, which translating that, multicellular. Now, multicellularity evolved multiple times. We can tell that by looking at the tree of life, who's related to who, and which lineages are multicellular. And we can draw parsimonious conclusions, the simplest hypotheses about where multicellular life began. Now, multicellular life is only possible when cells that are dividing remain glued together to be part of the same organism. And this requires several aspects, several characteristics to originate. Cooperation. The cells that are dividing need to be cooperating with each other, helping each other. We are cooperatives of trillions of human cells, animal cells. Specialization. that is an outcome eventually. Now, initially, something might be multicellular, but not have different specialized cells like some of the algae, like red algae, green algae. They're all mostly just the same kind of cell. And as I've already emphasized, multicellularity has evolved dozens of times. So cooperation early on, followed by potentially specialization. Unicellular slime molds, those are depicted in the middle part of your slide, those stalks. Dictyostelium discoides, it's the species name. These are single-celled unicellular creatures, things we typically call protists. But under certain environmental conditions, these single cells can glom together and form slug-like masses, multicellular slug-like masses for making spores, for reproducing. So there's a glimpse in the protist world into how something can transition unicellular to multicellular. Interesting. There's some fossilized scalloped discs down on the very bottom of your slide. They're not very big, but they appear to be multi-celled creatures. The thing is, we don't know what they were. And they show up for a while in the fossil record then, and then they disappear, almost certainly went extinct. An early experiment in evolution for multicellular life, we may never know. We start seeing filaments of organisms fossilizing 1.6 billion years ago. Just little strings of cells that are stuck together. Red algae. There's a red algae depicted in the bottom right-hand corner. You can see a size scale there for how big it was. 1.2 billion years old. Green algae, not depicted here. 750 million years old. The image on the very top of your slide is an image of bacteria, which can form biofilms, aggregations of lots and lots of bacteria, which are cooperating and helping each other, and in some sense, acting like a multicellular organism. So these are examples of the origins in multiple places of multicellular life and what it takes to be multicellular. So here we have the dawn of the animal kingdom, because at some point we see the origins of our own animal lineage, Animalia or Metazoa. Now, sponges arguably are the simplest animals around today. So we might use them as a model for what the earliest animal life forms were like. Indeed, we do have evidence of fossilized sponges going back 650 million years ago, and that's shown in the top figure. Figure B shows a fossil. Figure A shows living sponges. Now, we have cholesterol biomarkers that are unique to animals. And those show up just as molecular biomarkers at 635 million years old. So that tells us that there had to have been some animal ancestors around back then based on those cholesterol biomarkers. Then we have trace fossils. And there are many other kinds of fossils we're leaving out of the story here that are animal-like. But trace fossils are not of the animal parts themselves, but maybe a burrow or some tracks, like little footprints. Those would be traces. Skeletons, maybe, traces. So we have trace fossils going back 585 million years ago, and they have to have been different from sponges because they are moving. They are locomoting. So perhaps they were worm-like, perhaps they were slug-like, mollusk-like, but something like this, perhaps, trace fossils. So for the animal fossil record, we have before the Cambrian period, which started about 540-ish million years ago, there was the Ediacaran fauna. That's what we call it, Ediacaran fauna. And it's named after a region in Australia called the Ediacaran region. And these represent some of the oldest fossilized animals that we have. Some other Ediacaran fossils have been found in other parts of the world, I think in China, but that fossil record begins 575 million years ago. Their O is for old. Some are obviously animal-like. Others are more ambiguous. The upper right figure on your slide there is showing this two-page panel from your textbook, and the left-hand page is showing Ediacaran fossils, some of which don't look like any animals on Earth today. And then the right-hand panel is showing a little bit later the Cambrian fossils, part of what's called the Cambrian explosion. So many of those Ediacaran fossils disappeared by about 540 million years ago, and then the Cambrian animals took over. 4 stages of Cambrian fossils. Now the chordates, which is what we belong to, fishes, amphibians, reptiles, etc., we see the earliest chordates about 515 million years ago. So the beginnings of our own animal lineage that we belong to. Many went extinct. For example, trilobites went extinct about 250 million years ago, so they existed for a couple hundred million years. Lots of lineages originated and then were snuffed out by extinction. Now because, remember, Lagerstätten, this Burgess Shale, for example, that's up in British Columbia, shows us this Cambrian explosion of animal fossils, and we can reconstruct whole ecosystems and understand what lived back then. So now the image on the very bottom is showing a very early chordate-like creature from 515 million years ago. And then tens of millions of years later, we have fishes swimming in the ocean with fins and fierce jaws. So lots of transformations were always happening. So as you can see, life originated in the sea, in the water, and much of life still remains in the sea, marine life, freshwater life. But clearly some life forms made it ashore and adapted to life on land. There were several invasions of land. So going back 2.6 billion years ago, we can see fossilized evidence of terrestrial microbial mats. So bacteria, archaea. That's all that was living on land. Otherwise, our land masses would have looked barren of life. We see spores that are plant-like spores, 475 million years old. And those indicate the earliest plant life moving onto land. And those spores are most similar to liverworts, which I don't know if you've learned about those, but liverworts are depicted in figure B on the right-hand side of your slide. So we have plants today called liverworts, and these fossilized spores are very similar to liverwort spores. The oldest fossils of fungi are 400 million years old. And today, fungi are very closely related with plants, symbiotically, like mycorrhizae, for example, you might remember. And we have evidence that the plant-fungus relationships go all the way back to when plants were invading land with 475 million years ago. Animals began, we can see animal tracks that were 480 million years ago, little tracks of footprints on a beach-like dune, most likely an arthropod. We don't know if it was truly terrestrial or something that lived in the water and just happened to be walking, on a sand dune. But the oldest animal is a millipede, shown in the bottom left, about 430 million years old. Vertebrate tracks show up at 390 million years old, and the oldest tetrapod, land-dwelling vertebrate with limbs and fingers and toes, about 370 million years old. There's lots of dates to remember here. Yes, I want you to be familiar with these dates because these are giving you a record to remember the history of life on Earth, which is important knowledge for understanding evolution at many levels. So we're wrapping up here with who are some of our more recent arrivals. So many groups that are common today, extant living species, indeed evolved relatively recently. They haven't been around a long time. Now, of course, they have connections to ancient ancestors, but the lineages around today are relatively young. Synapsids. Look at the image in the left part of your image collection there. It shows almost a dinosaur-looking creature, but that was not a dinosaur. That was a synapsid. That was actually an animal that's more closely related to mammals and is very, very distantly related to dinosaurs. But these creatures lived about 280 million years ago. Mammals didn't show up until 150 million years ago, and mammals evolved from these creatures. So mammals are somewhat recent arrivals, but 150 million years, still quite a chunk of time. Dinosaurs show up in the fossil record about 230 million years ago, and they dominated our planet in terms of large animals until 65 million years ago, after the Great Cretaceous extinction. So that was quite a long period of ruling the earth. Mammals were not very abundant during that period of time. Flowering plants, now the dominant plants on earth, show up about 136 million years ago. Again, it's not that long ago in the big scheme of things. Grasses originate about 70 million years ago. Think about all the fields and the plains of grasses on Earth today. Grasses are a dominant plant type, and they didn't arrive until 70 million years ago. And they weren't even really common until about 20 million years ago. So grasses are more efficient at extracting carbon dioxide from the atmosphere, so they have come to be very dominant plants. Major mammal lineages pop up about 50 million years ago. That means primates, whales, bats, rodents, many of the major lineages 50 million years ago. Not in their present form, but early members of these lineages we can tell are related. Sahelanthropus, that's that ape-like looking creature on the right-hand side of your slide. 7 million years ago. It's one of the earliest pieces of evidence that we have of our own human lineage, separate from chimpanzees and bonobos. So 7 million years ago, not that long ago. Quite a history we are unraveling